Alrighty, ladies and gentlemen, in this video we're going to have a pretty exciting, information-packed little episode here, but we're going to talk about nerve conduction. So there's two types of nerve impulses. There's action potential and graded potential. Action potential is long-distance signaling, where graded potential is short-distance signaling. So there's short-distance signaling, there's graded potential. It's really all we're going to need to discuss about that right now. We're going to focus on the action potential. The action potential, a specific type of nerve impulse. So action potential definition is a rapid reversal of membrane potential, but with the F is membrane potential. So membrane potential is the separation of charge across the cell membrane due to the unequal distribution of ions. So ions, this action potential kind of works like electricity, if you will. So this unequal distribution of ions causes a separation of charge and causes these nerve impulses, aka this action potential, this somewhat system that really does function like electricity in our house, and we're going to look at that right now. So membrane potential is that separation of charge across the cell membrane due to the unequal distribution of ions, where that action potential, just mentioned it, is the rapid reversal of the membrane potential. So this rapid reversal of the membrane potential, it does cause that electrical impulse to go to a muscle, to go to a different neuron, to really control all of the functions in our body. And we're gonna see in a video or two how this really all works together, and it's pretty freaking cool stuff, to be honest with you. Like, I don't care if you like science or not, it's amazing how all these actions and functions in our body take place just within milliseconds. We don't have to think about it, but it's such a complex process. But that's neither here nor there right now. We're gonna get to that in a minute. But so membrane potential. So every cell does have a membrane potential. If there's, let's just theoretically say, a negative charge or two negative charges on the inside and two positive charges on the inside of a cell, where there's two negative charges on the outside of the cell, two positive charges on the outside of the cell, the membrane potential is going to be 0. So this specific cell would have a membrane potential of 0. However, if with a cell that has three negative charges, if you will, on the inside of the cell plus one positive charge, however, if there's three positive charges on the outside of the cell and only one negative charge, there will be a membrane potential as well. So there is a way to actually calculate the membrane potential, which is beyond the scope of these videos. If you want to look it up, it's the Nernst equation, pretty complex equation, but that's how membrane potential is calculated. We're going to focus on other aspects of the membrane potential right now, though. So the membrane potential of a resting neuron, just a neuron that's at rest in our bodies, is negative 70 millivolts. So millivolts is just the name, or the form of how we measure action potential in the body is millivolts. So if you think of like the volts or volts on your amplifier, on radio, or any electrical system, that's very, very similar, except we're talking about millivolts, which is a very, very, very small electrical charge here. So the 70 portion of the negative 70 millivolts is the identification of the degree of separation of charge. So this is how far the charge is separated across the cell membrane. And then the negative or the positive, so in this case the negative of the negative 70 millivolts, identifies the primary charge inside the cell. So inside the cell of a resting neuron is going to have a greater negative charge than positive charge. Hence, why the resting neuron has a membrane potential of negative 70 millivolts. So negative 70 millivolts. Make sure you know that. We are going to work with that in the actual next episode here, next video. But resting neuron has a membrane of negative 70 millivolts. So the millivolts is the label given to membrane potential. The 70 is the identification of the separation of charge, and then that negative symbol is the primary charge inside of the cell. So every cell in the body does have a membrane potential, even if it's like in the previous example 0. However, muscle cells and neurons are excitable. So excitable means that they can change their membrane potential. For instance, like epithelial cells, they can't change their membrane potential. What their membrane potential is what it's going to be. It can't be changed. However, with neurons and muscle cells, it could change the membrane potential, and that's what ultimately causes these electrical impulses to be sent. And the ability to change is due to gated ion channels. So gated ion channels, it allows. Well, we're gonna look into that right now. So there's inside the cell, there's less of a concentration of sodium compared to outside the cell. So when these ion channels open of the cell, due to facilitated diffusion, the higher concentration of sodium outside the cell moves into the cell in what's called a sodium influx. So due to the concentration gradient, the sodium moves into the cell, and also due to the electrical gradient as well too. But the reason why there's a negative charge of a resting neuron is because those well, not I guess as much for the resting neuron, but we're talking the gated ion channels right now. So the sodium rushes into the cell due to the concentration gradient as well as the electrical gradient. So for instance, with the resting neuron, let's say with the resting neuron, obviously the membrane potential is negative 70 millivolts. If the membrane potential moved to negative 50 millivolts, just to give you an idea, the membrane potential would actually decrease. Since there's less of a separation of charge, it's moving closer to 0. And since it's becoming less negative, the membrane potential is decreasing. So it's somewhat confusing when we're working with negatives, like, wait, we're having less negative, so it's becoming less negative, which means the membrane potential is decreasing. However, if it moved to membrane potential moved to negative 90 from negative 70, the resting neuron, the membrane potential would increase, put it that way there. But from negative 70 millivolts to negative 50 millivolts, the membrane potential is decreasing. So if these, just to really reemphasize the fact of these gated ion channels, so if the sodium channels open, there's going to be a sodium influx, and that's due to that concentration gradient as well as that electrical gradient. So I've said about 2 times the SAR, but that's very, very important, and it's going to be especially critical in this next video when we tie it all together. So that sodium influx due to, you got it. So a cell can depolarize, it can repolarize, and it can hyperpolarize. So there's definite definitions for each of these, and they do make sense once we start discussing them here. So depolarization is when there's less separation of charge. It's when when the number of the neurons millivolts are moving closer towards 0. So in the example of negative 70 millivolts to negative 50 millivolts, this would be a depolarization. There's a less separation of charge. So obviously 50 is less than 70. It's going to be less separation of charge, moving closer towards 0. That's depolarization. Less separation of charge, the number's moving closer towards 0. However, repolarization is when the membrane potential of a when the original resting membrane potential of a cell returns to its original number. So repolarization is when, say, a cell, as we see, moves from a plus 30 millivolts membrane potential back to a negative 70 millivolt resting neuron. So repolarization is when a cell returns to its original resting membrane potential. So it's just going back to its resting membrane potential, and with the resting neuron, that'd be returning back to negative 70. That would be repolarization. However, hyperpolarization is pretty much the opposite of depolarization. It's moving some more. those notifications, they get in your head. They do. They mess with you. You gotta focus. So hyperpolarization, pretty much the opposite of depolarization. It's when there's a greater, the separation of charge increases, and the number, the millivolt number, the membrane potential number moves farther away from zero. So in the instance of negative 70 millivolts, the resting neuron, if the membrane potential moved to negative 100, for instance, let's just say, that would be a hyperpolarization. So there's a greater separation of charge, and the membrane potential is moving farther away from zero. So there's a lot of information in this video. Feel free to pause it and just work back with examples. If there's still any uncertainty, just type in the comments, and we'll delve into these concepts even deeper. But I mean, really just got to work with them. Work with repetition, and they all will start to click. There's a lot of big words that really mean simple things, but action potential is a rapid reversal of membrane potential. Membrane potential is the separation of charge across the cell membrane due to unequal distribution of ions. All cells have cell membrane or a membrane potential. However, muscle cells and neurons are excitable, meaning they could change their membrane potential. Where like epithelial cells, they can't change their membrane potential, but the changing of the membrane potential is due to gated ion channels. So, for instance, sodium can influx into the cell, which which is due to the concentration gradient, also due to the electrical gradient. And then depolarization is when there's the separation of charges is less, so there's less separation of charge. The numbers moving, the membrane potential is moving closer to zero. Hyperpolarization is the opposite. There's an increase in separation of charge, so the membrane potential is moving farther from zero, where repolarization is the return to the original resting membrane potential. So in this next video, we're really going to piece it all together and just see how it works in our body. I mean, we wouldn't be here without it, put it that way.